Hello and welcome. You are listening to a podcast by the Milner Centre for Evolution at the University of Bath. I'm Professor Turi King, your host, and today I'm talking to Zaman Iqbal, Professor of Algorithmic and Microbial Genomics here at the Centre. Zam uses computers to look at the genetic makeup of disease-causing bacteria to understand how they evolve and how they're becoming resistant to antibiotics. Zam, lovely to chat to you. I want to start with the fact that I know you have a slightly unusual career history, so tell me about that.
Yes, I do have a slightly long and winding road to get here. I started out as a mathematician once upon a time. I did a degree and PhD in maths, at which point I just decided I was really tired and needed a change. So I went and worked in the software industry for about 8 or 9 years. I worked in mobile phones. I worked on the first operating systems for the first phones that had cameras in them, about which I was very, very skeptical because I didn't see any reason why anyone would want cameras in their phones. So, anyway, luckily I was wrong about that. And at some point, about 8 years in, I realized I was really missing science and started learning about biology again, which I'd been interested in as an undergraduate, but I'd never really followed up, and started looking for whether or not there were jobs where I could contribute. I thought at the time that maybe the right thing to do would be just be a programmer helping biologists. And so I got a job on something called the 1,000 Genomes Project, which was just when Illumina technology was taking off and they were going to sequence 2,500 human genomes. I basically got to sit in rooms with a lot of famous people and listen to them debate about things and learnt about basic algorithms people use to study genomes.
So what were you working on?
Well, at the time, our idea was, and this is not me, this is a global consortium, the idea was to sequence 2,500 human genomes, which was a massive statement back then. We had 2 human genomes up to then. And the idea was to try and understand the genetic variation that we all share and which separates us. So we took 2,500 people from 26 different populations, sequenced their genome in a certain sense, learnt about the common variation between them all. And one of the funny things about how we study genomes is we try and describe everyone in terms of how they differ from a standard. This is the standard human. And, Turi is just like the standard, but she differs here and here and here. Now that works if everybody is very close to the standard. But in some bits of the genome, that just doesn't work very well. It's a bit like describing some route around the London Underground in terms of always starting and ending on one particular line. Sometimes there are chunks of DNA which are very different between you and me. And sometimes it's just not helpful to describe that in terms of a reference. So I started thinking about ways to compare people's DNA directly instead of introducing a third party saying, Turi and I both differ from the standard in this way. We could say, how do we differ from each other? And I developed some methods to doing that and looked at the human populations we've been sequencing. And, we discovered that there was quite a lot of variation that was completely missing from the reference. You wouldn't know it was there, but which did differ between people. And the most obvious stuff is to do with the immune system. And that wasn't a surprise. People would have expected that. And that led me down a path towards other species, to be honest, because I got excited about weird and wonderful ways that genomes evolve. And humans are fine, but there's so much more other stuff going on in other species, and particularly in bacteria. In fact, your and my genomes are basically 99% the same. Even if we differ at some mutations, we have the same genes, we have the same chunks of DNA. But if I took 2 bacteria from the same species, they might only be 50% in common. And they have huge amounts of what we call mosaicism, so they can absorb lots of different blocks, and you make this kind of patchwork quilt of a genome. And that's super interesting.
So you come back into academia, but what leads you down to the path to the research that you're doing now?
Well, I guess what I did was I was trying to find ways to understand how different genomes vary. And sometimes with these things, you need to frame the question the right way. If you frame it the right way, everything becomes a lot easier. So the way we were framing things at the time was describing everyone in terms of how they differ from a standard genome. And that works when everyone's very close to a standard genome. But when they're not, actually it becomes easier to compare everybody against everybody. So I developed algorithms and methods for building a kind of, what we call a pangenome. So something that represents all of human genetic variation, and then using that as a structure to look at all of the variation within the species. And that was super interesting for humans. So we built the first human pangenome like more than 10 years ago now, and looked at how which bits of the genome were varying in different populations. But that whole methodology was super interesting. And actually, the concept of a pangenome came about 10 years before that. It was discovered in bacteria. People were trying to develop vaccines, and they thought the sensible thing to do was look at the proteins that would be generated by a particular bacterial species. And then they were a bit shocked when they took a few genomes from that species to discover that they were not as similar as they expected. They expected everyone to have more or less the same genes, but actually it turns out that in bacterial species, one of the ways in which they're able to adapt super quickly to new environmental conditions is passing huge chunks of DNA around between each other. And those chunks of DNA code for entire functional units that give them new, I keep saying superpowers, sorry, it's a bit childish, but you might be able to metabolize something new, you might be able to photosynthesize, they do crazy things. You can fire harpoons at adjacent bacteria and blow them up. You can release toxins, all kinds of things like that. And those are all passed around very quickly. And so the pangenome of a bacterial species can be 10, 100 times bigger than the actual genome of any individual bacterium.
And that's really important because, as you're saying, bacteria evolve really quickly, and they're slightly weird in terms of evolution. So the way we think of evolution normally is like, we pass down half of our DNA from each parent goes to each child. It's about going down through the generations. But bacteria are weird in that they can swap bits of DNA back and forth between one another. So they're unusual that way.
You're right. Every bacterial cell has two types of DNA. It's got the chromosome, which is passed down to their children, and that is inherited sort of vertically through the family line. And they have other DNA. Sometimes it's actually specifically separated into circular rings called plasmids. And they may get passed on to the children, but they also may escape the cell, move to a different cell. And so now their evolutionary history has moved through unrelated cells, may move to a totally different species. So that means very rapid and unexpected things can happen. Maybe it doesn't work. In the new place, maybe that new bit of DNA is unhelpful and that cell will die. But sometimes that's the chance for incredible novelty, a new function that wasn't there before, and so on. So there are lots of bacteria who have acquired the ability to move between motile and sessile, or can photosynthesize, or can kill things, or have an immune system. All kinds of things like that are passed around very quickly, sort of sideways.
Yeah. So I'm a human geneticist, so I know the very basics really about bacteria, but I know that plasmids are really important. So what are plasmids doing? I mean, I like the fact that you kind of get these like patchwork quilts of plasmids, because you get one bacteria going, ooh, I have this snazzy little superpower. I can do this. Here, you can have it. So what are plasmids doing?
Well, that's interesting. They do all kinds of things. They seem to mostly provide functions that are locally adaptive. Mm. So they're an advantage temporarily.
Yep.
If they were permanently always needed, then I think generally you would find the functions would be absorbed into the chromosome and they would be there forever. So what are the challenges bacteria face? They've been around 2 billion years. The things that they've always faced are viruses, which predate on them, and each other, and things like temperature and environment. So plasmids provide a variety of functions. If you look across a generalist species, you will find a wide variety of different plasmids, in the different cells. You can think of that as a kind of bet hedging, I think. If there's some plasmid that's super critical, even if you lose some members of the species, that the others will survive, and also potentially that plasmid could spread again. So that's the kind of thing they do. I mean, we notice them and care about them a lot at the moment because they often carry drug resistance, or they carry genes that allow the bacteria to survive when they're treated with antibiotics. That makes a big difference. And since we've introduced antibiotics, they've spread super fast across the world. And often people who are genomic epidemiologists, so people who track down how the virus is spreading, if you're in a hospital and you've got several people who are ill and they've got the infections resistant to a certain set of antibiotics, then you might think, okay, well, is there a single bacterial species that's spreading? Is there something that's very fit and it's moving through my ward? Or sometimes what happens is there's actually a plasmid that's moving between different species. There are very famous examples where you have, more than a decade of an outbreak in a hospital, and the thing that is in common is either a plasmid or even something smaller than a plasmid jumping between plasmids. So, yeah, it's chaos.
Yeah. So what are you doing?
I try and invent ways to think about these things. And if you find the right way to think about these things, everything becomes easier. And ways to think about things means computer programs. So like a simple question would be, if two people are ill and they both have E. coli, is this the same E. coli that's infected them both? Maybe they share the same infection. And that's an easy question to answer because we can look at the sort of family tree of the bacteria and try and see how closely related they are. And if they're very, very closely related, almost the same, then it becomes quite credible that actually there's been an infection passed between them. Whereas if they're like thousands and thousands of mutations apart, well, there's no way that happened within the 2 weeks that they were both in the same ward. So they've got two different infections. That's sort of well understood. But with plasmids, because they actually change their genomes very dramatically, they will eject chunks of DNA, they will rotate bits around, they will jumble things up. It's a bit like the ship of Theseus or Trigg's broomstick, if anyone remembers 1980s comedies. It's hard to know if these two plasmids are the same plasmid. Is it credible that this one has changed, thrown out 2 kilobases of DNA and gained another 5 kilobases of DNA? So what we look for are computational methods for looking at 2 chunks of DNA and saying, well, given how plasmids change, which is certain kind of structural changes, is it credible that there's not too many steps to go from one to the other? And sort of inventing ways to do that, it's sort of one of the things we've been working on recently.
So it's like you're kind of tracing sideways family trees almost of plasmids and looking how they are chatting to each other and swapping bits and how they're related. And I'm guessing, given that you're using these huge kind of computational methods to kind of interrogate these plasmid genomes on a really big scale, you're looking, is it globally now that you're looking?
Yes, we look globally. I mean, one of the things we're trying to do is, if you're trying to look at how plasmids or other things move across the world and across species, it's not enough to just, get the bacteria from your fridge or from your local hospital. You want to look very broadly. And you can't do that yourself because even if you exclude the expense, I mean, where would you sample from? It's actually much more effective to look at everything that everyone else has ever sequenced. So there are huge DNA archives where people share DNA of bacteria that have been sequenced. So in principle, we can just go and look in there and see, where are these plasmids? The trouble is that the process of constructing a genome from raw experimental data is imperfect. And the data that's in the archives is sort of a hodgepodge of different methods, of different quality control systems. Most commonly, you find a super exciting thing and actually, well, either it's an error in your code or, all of these things that you think are exciting, they were all done by the same person and they all made the same mistake, or they've done something slightly unusual. So what we've done is we've started a large global consortium of people taking all of the DNA of bacteria that have ever been sequenced and reprocessing it all, building high-quality genomes. And yes, we will be looking through all of that, all the plasmids in that. That's quite exciting. We've got 2.5 million genomes at the moment.
And I know one of the things you're looking at is antimicrobial resistance. So we've touched on already the fact that bacteria are now becoming resistant to antibiotics. And that's something else that you're looking at the moment, isn't it?
Yeah, that's an ongoing theme. So one of the big challenges for us is how to deal with infectious diseases, and treatment of infectious diseases is actually an evolutionary challenge. So it's not like a kitchen where you just clean it and the bugs go. When you treat a patient with a drug, the bacteria inside the patient are growing, they're reproducing, they're mutating, they're evolving during the therapy. So during the first ever clinical trial of streptomycin for tuberculosis, and during the trial, patients got better and got worse because there was resistance that evolved within the patients during the lifetime of the trial. And that's an ongoing problem for us. It's a problem per patient because we want them to survive. And it's a problem across the population because small numbers of bacteria can survive that therapy and spread. So for tuberculosis, it's a problem because a lot of the people who get ill from it are poor. They don't have access to healthcare. They live in greatly overcrowded conditions. And you can be infected and be asymptomatic for a while. You don't know that you're infected. Actually, I did a back of the envelope calculation and I thought 6 months in, you might have over 1 billion bacilli in your lungs, which means actually, every position in the genome could have mutated.
Wow.
So if we assume that drug resistance can be caused by a mutation in the genome, that sort of means that every type of resistance is already there before you start therapy, which is why standard therapy for TB is actually multiple drugs at the same time. It's much harder for the bacteria to develop resistance to multiple things at the same time. So this has been an ongoing challenge for decades. And the problem for us is that we develop new drugs at a very slow rate. So it's exciting times in TB in some ways because we've discovered that we can have slightly shorter therapy regimens that are just as successful, but we don't have an armory of new antibiotics ready to treat. So it is inevitable that there will be resistance to any antibiotic that we generate. And so there's not a very good answer to that, apart from the fact that we need very good diagnostics.
So is your work trying to work out what the genetic changes are that are making this bacteria resistant to the antibiotics? And are you trying to kind of like track those changes, I suppose, kind of through time and geographical location? Because once you know about that, if you kind of go, ooh, okay, they've got this particular version of the bacteria, that's going to be resistant to this antibiotic, hit it with this one. Is that the kind of stuff that you're trying to do?
That is the kind of stuff we do, yes. Basically, there are drugs where there's a lot of resistance out there already. And for those, you can try and work out which mutations cause resistance by a statistical process. Collect a lot of samples from patients, measure them, mix the bug and the drug and see if it kills it, essentially, and measure the level of resistance in them all, and then get the genomes for each of those bacteria. And then each of those genomes, you can break that down into which mutations they have, and then you can work out which mutations correlate with resistance. That is not a totally trivial process. So we spent 5 years actually collecting about 15,000 samples from 23 countries, measuring drug resistance to 13 different drugs. That's a consortium called the Cryptic Consortium. And what we did manage to do is actually build very comprehensive catalogues of resistance mutations, which got absorbed by the WHO. The problem is, with new drugs, you want to know as quickly as possible what's causing resistance. But if you want to spot that fast, you don't have many samples. And if you don't have many samples, you don't have much evidence. If I got a new TB sample from a patient, there would probably be 1,000 or 2,000 mutations in it. And which one is causing the resistance, I wouldn't know, not up front. So one of the things you can do is go to places where they first started therapy. So bedaquiline is one of our latest and greatest drugs. And I say latest and greatest drugs, it went into use 20 years ago. And go somewhere where they started treating them earlier than the rest of the world, so South Africa, and track resistance that's appeared there. And so when we did and when we went to look, essentially there are two options. Either we would see one strain or a small number of strains of TB that were somehow becoming very successful and spreading, and we'd see the same thing in many, many patients, and that's not what we saw. Or we might see drug resistance evolving in slightly different ways, again and again and again, in different sort of genetic backgrounds. And that is what we saw. And so that means that that pressure to evolve the resistance is there again and again in different patients. We were using data from during the pandemic, and there's more data has come through since then. So we're going to track forward and try and understand whether or not anything specific is spreading or what's going on.
So what's the hope? Is the hope that eventually it might be something that clinicians can use very quickly in the clinic? It's like, right, we're going to get a sample here. You've got this particular bacteria. It's got this particular sequence. We know not to bother hitting it with this. We're going to hit it with this. Is that the hope that eventually it'll become something that clinicians can use pretty quickly?
Yeah, that's absolutely right. So there are two things you need. You need diagnostics to tell you what to do, and you need therapies. And what we're talking about here is the first half. So if you want good diagnostics and maybe to tie in with epidemiology to know who's infecting who, then whole genome sequencing offers that if it can be made rapid enough and easy enough to use. And so there's a strong push to do all of that. I mean, you need everything from how to get the sample from the patient prepared in the right way to go in the sequencing machine. Actually, that's the hardest bit now. The algorithms and the computational bit is all reasonably straightforward now, especially for TB. So the tricky bit is getting sputum from a patient. Most of that is human cells, which we're not that interested in sequencing. So you need to sort of narrow down to the bacterial cells and sequence just those.
So what's next for you?
Well, I guess there are two major things that are happening in the field technologically. One is the volume of data that we're generating keeps doubling, and that presents big challenges for computational methods, especially if, as like me, you're interested in scanning through all of the data that's ever gone before. Quite often you want to see, has this been seen before? Is this a strain of E. coli that's been seen before? Is this a plasmid that's been seen before? So you need kind of search indexes. So we develop algorithms to do that, but it's very hard to keep up with data that keeps doubling. So definitely working on that. And the other thing is that sequencing data itself is improving. And in our search for antibiotic resistance genes and tracking them through bacteria, we often find that bacteria, they're not satisfied with just having one copy. They'll double up or triple up. They'll have lots of copies in their genome sometimes, and they copy-paste themselves in funny ways. And because they've copy-pasted themselves in funny ways, it actually becomes harder for us to reconstruct the genome. If you've got a jigsaw and more and more and more of it is copies of zebras, it becomes harder and harder and harder to work out, which zebra goes where. But it's actually often quite important to know, not just where the zebras are, but almost like what their identities are, because different versions of an antibiotic resistance gene can be subtly different. So we develop algorithms to try and do all this jigsaw work. And it turns out the new technologies are actually much better for that. So that's pretty exciting.
So what's the ultimate goal of your research? So what would make you happy if you get to the end of your career and you think, I did that. That's what I've been trying to solve. Is there something?
I can give you an answer now. I think it's probably true that if you asked me every year over the last decade, I'd give you slightly different answers every year. But right now, I think if we could come up with a way of thinking about evolutionary trees of bacteria and the evolutionary trajectories of plasmids together, I think that would be amazing.
Zam, thank you so much for talking with me. This was a podcast by the Milner Center for Evolution at the University of Bath. I'm Turi King, and thank you for listening. If you have any thoughts or comments on this or any other episodes, please contact us via our X channel at Milner Center. For more information about the Milner Center for Evolution, you can visit our website.